So delighted to welcome Brooke, Mangi, Ethan, and Mike to the panel discussion today. So the kind of two main themes or topics that we'll kick off with are around standardization, and the second will relate to VOCs and healthy human subjects and building a VOC atlas. We also want to make this a very interactive session, so the participants, if you have questions related to those two topics or questions, anything breath related, please feel free to post them into the Q&A and we'll be sure to direct those to the panel members. So to kick it off, we will get the panel members to introduce themselves. And Mike is joining from yesterday. So Mike, maybe we know who you are, but maybe if you can give just a quick 30 seconds recap, please.
Sure, thank you, Billy. So I'm Mike Wilde. I'm a lecturer at the University of Plymouth in analytical chemistry. And yeah, I've done several years of breath research.
Brilliant, thank you. And Brooke, maybe we could move to you next. I don't know if you have a slide you'd like to share to set the scene a little bit for you.
Sure, yeah, I can do that. Let me so it's kind of focus more on our kind of normal breath work that we're doing right now, but I can introduce myself and put this up at the same time. So I'm Brooke Kaiser. Nice to see everybody, not really see everybody, but have all of you out there. I'm a biomedical scientist at Pacific Northwest National Laboratory. So we're in Washington State in the US, and I have been there for about 13 years. My background is microbiology, but have gotten into VOC work and looking at human breath over the last about 5 years. So one of the projects that we have going on right now focuses on looking at healthy human breath, really with the goal of trying to figure out what that baseline of normal breath looks like for a healthy population, in order to then build upon that later for breath-based diagnostics. So understanding what's normal is, we think, pretty critical to understanding what abnormal looks like. So this just talks you through a little bit about what we're doing. We have a pretty big cohort of individuals that we've used for breath sampling. We analyze those samples on a two-dimensional GCMS instrument, and you can see the names of the instruments there. We'd use two instruments for complementary separation, try to get the most diversity of those VOCs that we can. Then you can pop down to the data analysis part, which we generate hundreds of data sets. We align those and start to look at the information about those cohorts in order to figure out, instead of what's different about individuals, what are those things that we all have in common? And then our last step, of course, is to really dig into the statistics, really dig into the metadata that's associated with those individuals to try to say, okay, maybe we have a cluster of people who are healthy. We might have some subsets within that show some similarities on their demographics. Maybe it's smokers, maybe it's individuals taking a certain medication, and try to identify those features that really are influencing breath, as well as those people who we might think are more normal and then they kind of separate out. So just really trying to understand on a global scale, what normal breath looks like.
Fantastic, thank you very much. Mangi, maybe you could introduce yourself and share a little bit about the work that you're doing and the approaches you're taking.
Yeah, and I also have a slide.
Perfect, thank you.
Yeah, so this is Mangilal Agarwal. I'm the professor of mechanical engineering and director of Integrated Nanosystems Development Institute here at Indiana University in Indianapolis. Basically, long story short, my work, I'm a sensor person, and my research focus around the sensor and sensor development for healthcare applications. And back in 2013-14, we came across diabetes alert dogs that can smell a person and alert them about hypoglycemia. So as an engineer, I was intrigued to see what dogs are smelling, and if we can identify what they're smelling, then we can develop a portable device So we started looking at sample class where the dogs get trained. So it was either the breath or the sweat. So we collected those breath and sweat samples and ran it through GCMS, got the biosignature, and started developing the sensor technology for those biomolecules, especially the volatile organic compounds in breath. Moving forward, we also work with Brooke and Ethan here on healthy breath study, where we are trying to find a class of VOCs that are present in general breath, healthy breath, and see what's the variation, what's the range of those VOCs, and try to identify a kind of database of VOCs that can correlate to healthy subjects. Now we have other studies on prostate cancer. We have studies in cystic fibrosis. We ran some COVID studies. So having this database will also help to correlate how a disease profile will look like and how we can distinguish the VOCs that we found in healthy breath compared to other diseases. So our setup is very simple. We collect direct breath, expose it to SPME fiber, including the nanosensor array, and then the SPME fiber is run through GCMS, and we get the chromatograph and mass spectral data, and we then identify the VOCs that are present across. We collected about 150 subjects, breath samples, and then we are looking at also longitudinal, where a particular subject gives their breath for longer period of time and see the variation within the intra-subject and inter-subject. And here are some results that you can see. Now, comparing this with the disease state would definitely help to identify some of those VOCs that are prominent in healthy breath and also correlate to certain disease and what are the ranges. Yeah, the future goal is once we have that, then we can make portable devices that can be used in field for diagnosis applications.
Wonderful, thank you very much. And then lastly, Ethan, if you're able to introduce yourself, and if you have a slide to share as well, that would be fantastic.
Absolutely. Can everyone hear me?
Yep, we can hear you.
First step. All right. So hi, everyone. I'm Ethan McBride. I'm a scientist here at Los Alamos National Laboratory. I've been here for about 2 years. We've been in the breath field for approximately the same amount of time. I'm a part of the bioscience division here, and on a smaller scale, a part of the Mass Spec Center for Integrated Omics within biosciences. So we are sort of a multi-omics platform from metabolomics all the way up to proteomics. I've got a picture of our team here. For the breath research portion, we actually do direct analysis using secondary electrospray or SESI. You can see that in the picture there, and that's on a Orbitrap platform, so the Exploris 480. The front end that we have with the SESI also has ion mobility capabilities, so we're able to get all of that in a direct measurement. Most of our data analysis pipeline is sort of custom. Using exact math with breath data is sort of its own beast. And so we've basically combined our normal compound discover pathways for doing our database searches with some custom bioinformatics pipelines that we have here at LANL, and using those to talk to one another. Most of our data we try to sort of use with the standards set forth from the Metabolomic Standards Initiative that's in the bottom right. Just something you would normally see for a multi-omics pipeline. Yeah, so that's what we do here. Glad to see some direct breath measurement techniques today and yesterday. That's great.
Excellent, thank you so much. And Mike, we know the system you're using is GCGC mass spec. So I think what's been interesting, both from the introductions and the feedback that's come through from the poll, is the very broad range of approaches which are being deployed. And I think that is both on the sampling side of things, from different offline sample collection methods to online analysis, and then right through to the analytical techniques. So I think it's really interesting to see the range of different approaches being applied. And I think that then loops back into the first topic of discussion around standardization. And the question that we posed when we think about standardization, does this mean the choice of specific sampling hardware or analytical platform, or more to do with the procedures to ensure data quality? So how you ensure something is on breath, how you get definitive ID of that, and using this best practice or these best practices to be applied across different types of hardware. I think what was interesting from the poll results was a small number, but not insignificant, thought there should be standardization of the actual hardware itself. So I thought I might open the floor up a little bit to people to see what they think about that, because it seems the benefit of different technologies and approaches is that you can attack the problem on different fronts and get broader coverage of the VOCs that may be present. So I was going to open it up and explore that to start with. Maybe, Mike, you could kick us off, please. Should we try to standardize on the hardware, or should it be more about SOPs and best practice?
Yeah, so I fall very much in the latter. I think it's about best practice and SOPs and things, and how you approach the data handling. Standardizing hardware is an interesting one because most of us use the hardware available to us or that we're a particular expert in, and no matter how much you tell me the benefits of another technique, I want to pursue and see how far GCGC, for example, in my area will take me and what that discovers. And therefore, I think trying to implement, forcing people to use a certain hardware probably just won't happen. But by all means, unless that's not necessarily what the people that tick that box meant, they might have meant that where there are sort of set hardware, such as everyone generally has an Agilent GCMS in their lab, or other manufacturers available, when there's sort of universal hardwares, and perhaps there could be more standardization across that, perhaps.
I'll open it up to the rest of the panelists. Is there anyone, given you're all using very different setups, Mangilal, do you want to go?
Yeah, basically, VOC analysis is not new. We have looked at VOCs in environment, in soil for a long time, and people have developed standard methods for analyzing those VOCs and toxins, over time. But in those cases, basically, you are trying to find what you already know, and you can standardize the process based on that. However, in breath, right now, it's something like what we are trying to find in healthy breath is using different equipment, using different hardware, using different biopsy methods. What are the class of VOCs that we are looking for? And then once we identify a complete database, I think the next step is to see what VOCs are important for what condition, and then based on that, we can standardize the hardware if you're looking for a particular disease, and then particular VOCs, and you can standardize the hardware and the biopsy method for those methods, including in clinic, right? So you can have a standard process in clinic that is feasible for a particular medical condition, like if it is diabetes to it depends also on home-based methods. So it's not one standard. It depends on the application too.
And I think that point of what can be learned from other fields is obviously very important, very relevant to breath, and something that we'll touch on a bit further. Brooke, Ethan, anything you want to add on to the notion of hardware selection?
Yeah, absolutely. I think that, there's a long, strong history of people modifying instruments to try to analyze things that maybe haven't been done before. So, dream on, that's great. I think that there's a lot of strength to that, but certainly, at a certain point, being able to reproduce someone else's work is so important. And so having some of those standardized things so that somebody might be able to repeat it is absolutely important. So I would agree with Michael. You never know exactly what somebody could have meant by answering that question, but absolutely standardization is important, but being able to analyze things in a new and exciting way is always a good idea.
Fantastic. Brooke?
I'll just quickly, echo what they said, but also, excuse me, add on to that we, in our breath project with 3 different groups taking 3 different approaches, we've really seen a benefit in that actually, because I think when we come together and we say, oh, I found this, oh, I found that too, oh, I found something similar, it gives just greater confidence, I think, that those things really are real. And I think that a lot of different platforms can generate really, high quality data. And then what do you do with that data? So that is, another challenge as far as standardization of, like we've talked about a little bit, reporting and how you, do your data analysis is a challenge because we all have our own ways of thinking about that too. But I think that, yeah, spreading out different approaches only is gonna, I think, gain you confidence in what you're actually finding.
I think that's an incredibly important point. If you're able to make the same observation from different angles and different approaches, it is going to support that it is more likely to be a real finding as opposed to not. And maybe we could move on then and start with you, Brooke, pick up on the notion then of, if it's not about the hardware, if we all are going to be using different approaches to get broader coverage, etc., but we do want to try and get some type of agreement on how to do things. So things like reporting standards, how you deal with background contamination, ensuring you're IDing the compounds, etc. And maybe if we start with the contamination point, John Harshman earlier talked about the importance of background. So I was hoping we could start there and explore, if we think from a standardization standpoint, some of the thoughts around how to try and address that, given we are all using different instruments and approaches.
Yeah, for sure. We think about it. It's like your background, you're just going to have to deal with it, right? Like we're all breathing in the air, and there's just going to be some of that carrying over into our breath samples. The approach that we've taken for what it's worth is we've taken lots of background sampling. So anytime an individual gets their breath sampled that same day in the same room, we take a background air sample. So we have matching information. But instead of doing it one-to-one to try to like subtract out background in that way, we've kind of taken more of a, holistic approach, I guess, where we look at breath as a group. So lots of samples of breath versus lots of samples of background room air, and taken a statistical approach to say what's enriched in breath, what is present in breath and absent in, room background. And so there's a mix of features, of course, that fit into both of those categories. Mm-hmm. But that's kind of the overarching approach that we've taken to thinking about background. As far as the background from the room air, it's always instrument background as well, but that's kind of where we've started.
Brilliant. Ethan, any kind of insights from you and how you're currently trying to tackle background, be it ambient or instrumentation sampler?
Absolutely, yeah. I think I can tell horror stories about people putting floor polish in 6 rooms over, and we've been able to see it with the SESI. So those things are sort of unavoidable. I think it's really, really important to sort of understanding, whenever we're talking about background, sort of those endogenous and exogenous sources, and kind of gaining like a better understanding of where maybe some of the compounds that we're seeing are coming from. I know that we love to talk about that review paper from 2021 with all of the volatiles from breath. And a lot of those had never been seen before. So you've only got basically one reference there. So I think as we start to build out more and more papers, more references for compounds, understanding where they're coming from is going to give us a better idea of, are they endogenous, are they exogenous? And so at least on our side for the SESI, we've really had to pay careful attention to things in the room air, just like we do with some of our other analyses. And so yeah, you have to have a really strong understanding of what you're seeing and where you think it's coming from.
Fantastic. Mangi?
Yeah, I think Ethan and Brooke covered about the background, air, which is very, very important. It's basic principle to understand, like, when you are analyzing breath, what is in the background, and how the VOCs that you're seeing in breath, are they endogenous, and I think basically in our approach, we also collect background sample before and after we collect the breath sample from a subject. And then we are still trying to figure out whether we want to take a holistic approach or we want to take a basic approach of subtracting that background from individual breath on that day, of collection. So it's very important to understand, the breath itself with respect to the background.
Mike, anything to add there, or have we kind of covered some of the main points?
So we handle our room air background in a similar way to, as Brooke mentioned. However, we ask not just sort of in the comparison between breath and room air, but ask the same question of the room air data that you're asking of your breath data in terms of the diagnostic question, whatever you're doing. So it pulls up those differences that you can see. But I would also highlight the importance of just having that room air data. For us in a discovery study, you never rule out manual review, and so when you see outliers in your data set, just having that room air data can sometimes help explain those outliers. So if you see like a couple of patients with like massive IPA peaks or something, okay, one hypothesis for that is that it's the solvent in the room, or someone's used too much hand wash or whatever. But with the background room air, you can actually go back and check that, and you can be like, actually, yeah, that looks like it's consistent. There's a high correlation there between those two peaks. So there's this sort of this statistical handling, but just having that data available to you allows you to have that retrospective sort of scrutiny of the data. So if there is anything you decide to action, then at least you're actioning it on an informed decision and not just on a sort of guess or a priori knowledge.
And I think that's important because it relates back, some of the points that have been touched on, it relates back to being able to accurately identify what you're observing as well. And I think, again, if you look across a broad class of different approaches being used in discovery, there's some where you don't know what you're targeting. You're looking at an overall fingerprint like eNose, and then with more of the spectrometer-based approaches, mass spec and the like, it's even quite difficult in some of those approaches to definitively know what it is that you're seeing. So I was wondering if you could elaborate on how important you think it is that you ID in discovery, and if you believe that is important, what is some of the best practice we can think about there? And Mike, maybe I'll start with you. I know in your talk yesterday, it was great to see that you were reporting MSI level 1 type compounds in your work. So maybe we could start with you on this question.
Yeah, so the idea for us was really because we wanted to also try and move forward towards some sort of quantitative, depending on what level of rigor you're prepared to put behind your quantitative assessment. You really need sort of to know what sort of compounds you're looking at. So at least if you're using a reference chemical, you know if it's even similar to what you're looking at. But I typically would say it is essential to ID at the point where you're making decisions. So if you don't necessarily have to ID everything in breath. A lot of noise and stuff, it's very time consuming and therefore expensive to do. But as soon as you start making claims like markers, I've identified or anything like that, I think then it's important as soon as you sort of narrow down to features of interest or even features that you've removed as artifacts. Because equally, if you remove anything as an artifact, you should probably be sure that it is what it is before you do so. So I think as soon as you start making decisions with the markers of interest, then yeah, I think ID is critical.
Excellent. Brooke, maybe you could elaborate a bit further on that, describe what you guys are doing in your lab.
Yeah, very similar to what Mike just said, actually. We kind of take the stay blind to it approach at first. We look at a feature list, which maybe don't have names associated with them. And then when we start to do that comparison to narrow it down to things that are more interesting, then we start to give things names and follow up to really be confident in those identifications when you're actually saying, yes, this is this particular compound. So we stay blind to it for a while until we have a narrowed down list of what we really care about.
And Mangi, I think in your approach, you're using both pattern-based and spectrometer. Is that correct?
Yes. Yeah. So our GCMS is both MS spectrometer based. In that, I think we have to use basically what our approach has been. I agree with Mike and Brooke on this, that you have to look at the overall picture and then select features of interest and then run standards. Now there are only so many standards available that you can verify the ID. But then there are other issues with respect to standardization of data, which is problem with current spectral alignment softwares. So those needs to be also looked upon when you are trying to ID a particular VOC. The best way we feel is running a standard, but then that may also give you false data. So.
Ethan, anything to add? And maybe we could thread in lessons that could be learned from metabolomics, which is obviously very well established, and we've been building knowledge of biochemical pathways for the last 100 years.
Yeah, absolutely. We sort of have the advantage, I guess, with the Orbitrap platform of getting really good exact mass measurements. But obviously, we've lost some of the chromatography with the secondary electrospray front end. So we've tried to really build it out as whenever we're running our raw data through, say, Compound Discover and getting our database matches, taking that information and sort of correlating it with what we're seeing with our own bioinformatics pipelines and saying, these are compounds of interest. I've had my features reduced down. How good is that identification from the database? And then obviously, just like you talked about with the reporting requirements, most of what we do is at a level 2 for MSI. And that's, those are pretty stringent requirements. And I think that's a really, really good thing for sure.
And I think one of the points that was brought up earlier, it relates to background and endogenous and exogenous compounds. In the discussion I had with Sean, I think how we think about it here at Alstone is, first, you want to ensure that you're detecting something that's actually originating from breath and not the background. But something that's on breath can be of endogenous or exogenous origin. And actually, even if you look at some of the classic compounds like acetone and isoprene, they are abundant in the environment. Given it's important to ID those compounds, I'm wondering if you could comment on how easy it is to make a definitive assignment of this is of endogenous or exogenous origin. I know we'll lead on to that in the Atlas discussion, but we certainly struggle with that a little bit when we're looking at a compound and saying we know what it is, but where's it coming from? Maybe Brooke to kick us off on that one.
Sure. Definitely a challenge. So just say that up front. One approach that we've taken is kind of to take an additional breath based sample. So we're looking specifically at exhale breath condensate as well in our individuals. And that provides us with some protein information. And we hope that will give us a little bit of a leg up on trying to understand potentially metabolic pathways that are kind of represented by those proteins. Where we could see potentially secondary metabolites that are VOCs, or, they're not the fun metabolites usually that are in your KEGG pathways that are just easy to, check off your list. But our approach is, to try to take some additional complementary information to try to say, okay, do we see a biological reason why we believe this would be coming from a process within the individual endogenously versus something from the environment?
Ethan, anything to add on that from what you're observing on breath?
Absolutely. I think that might be one of the most important questions that we're faced with whenever we're publishing some of our work is you can identify something and feel very confident in it, and it makes absolutely no sense in the context of actually coming off of breath. I keep going back to the lit review paper. A lot of those that were in that lit review, which is a fantastic, by the way, absolutely do not make sense in a sort of biochemical pathway. So yeah, I think that's probably one of the most important questions we have to ask ourselves is, you know, if I'm seeing this, if I'm identifying it to a certain level, can I make sense of that? And that really is where the richness of this data is going to come from and allow us to start incorporating what we have found into new studies that maybe involve, you know, some more classical techniques, comparing what's in breath to blood and ISF and other things. So yeah, absolutely, I agree.
Mangi?
Yeah, I think my colleagues covered it all about, you know, what we call endogenous. But one thing I would point out is just because the VOC level is higher compared to the background, that doesn't mean that they are endogenous, you know, or you know, they can be byproduct of the food intake. So we kind of collect the food diary and all that to understand, you know, when we are trying to build this database with respect to endogenous and exogenous VOCs, where they are coming from. But yes, having a biochemical pathways are very important, but maybe there are secondary pathways that, you know, that's not in the literature or not well understood. So we have to identify those too. If we, like you said, if we really know what this is the VOC of interest, then we can do the reverse engineering to really find a biochemical pathway that may not have been already explored.
And Mike, how do you approach it? Obviously, yesterday in some of your great work, you've got, you know, you definitely know that's the compound that you're seeing. How do you relate it back to the biology and endogenous, exogenous?
Yes, so two things. Just to acknowledge that the challenge that Brooke sort of highlighted there is, so it might be put out there, but I don't think I've identified a VOC in breath that hasn't been reported in another source. So I don't think there is a VOC in breath that is unique to breath, just by the sheer nature of the VOCs that the sort of compounds that we're looking at. So you're always going to find another source. So that's sort of defining the challenge that you're up against with that, and then it's a case of justifying within that context, okay, what is realistically going to be influencing the breath for this person at that time, using the sampling technique that I'm using. One of the ways actually in highlighting the sort of the looking for networks in breath and correlated sets of features, one of the things that was very good at is sort of identifying features that probably aren't relevant, because we actually included some of them in the primary data when we asked the model to find us systematic differences between our groups. It found a cluster of molecules which were not from breath. They were quite clearly surfactants or emollients from makeup or contact with the face mask that we're using. But that's still we asked it for a systematic difference between, say, healthy and a disease state, and actually that makes sense in terms of that. If people are coming in healthy controls with makeup on compared to someone that's been in hospital for 10 days, whatever, then there's going to be differences. So, but looking at those groups where you have a compound such as like tridecanol, which could be in a makeup, but it could also, it's an aldehyde, so potentially of interest or whatever, where it correlates, if it starts correlating with those other compounds that just don't make sense at all, then it sort of suggests that it's maybe from a source that you're not interested in. And then when it doesn't, when it's sort of like some of the terpenes, a lot of them group together, and they're probably from, if you drink orange juice, you get limonene, but yes, you also get a whole bunch of other terpenes that will go with it. But if you get one or two terpenes that don't correlate, actually that's kind of interesting then. So is there something there? So we actually found the sort of the correlation networks quite interesting in terms of identifying those sources.
Yeah, that's fascinating. And it's interesting you mentioned the terpenes as well, because I think one of the eye openers for us a few years ago was the example of limonene, where it's like, it is definitively an exogenous compound coming in through diet, but it is on breath. And I think we used to immediately just kind of discard anything that we thought was coming from diet. But there's clearly a robust difference between those with liver disease and controls. And when you administer that that probe, it's going to be metabolized in the liver. So I think it's interesting actually, where if you try to shoehorn everything in to say it must be one origin or the other, actually it can close you off a little bit. Whereas just by understanding what's there, then you're at least able to build out from that and see, is there anything that's a bit unexpected that might have clinical utility? Mangi, did you want to have a follow-up there?
Yeah, Ethan and Mike, they both brought up good points, especially when you're looking at group of VOCs or the functional group of VOCs, then you can definitely correlate to endogenous versus exogenous, when you're looking the group like a signature of VOCs, right? But one thing that now we are talking about breath, but in our studies, what we also did was we collected urine samples and we are analyzing the VOCs from the headspace of urine and then trying to see what is present in breath and what is present, of course, different concentrations. But then we, in certain medical condition like prostate cancer, we have identified some VOCs that are present in breath are also present in urine headspace. Then we can confirm that, yes, this is through the metabolic process that is happening in the body.
Excellent. I'll take one question that's come in through the chat that relates to standardization before we move on to talk about VOCs and healthy subjects. So the first is, what are the thoughts of the panel about using something like the peppermint protocol as a benchmark for comparing different breath sampling techniques, and I assume analytical techniques as well? I'm not sure if everyone is familiar with it, but the basic idea is you administer a peppermint oil capsule and you look at the washout profile of the peppermint oil, and it has a mix of different terpenes and eucalyptol and those types of compounds there. Mike, I think you're familiar with that. Maybe you could kick us off on that one.
Yes. No, I'm familiar with the peppermint washout. Professor Paul Thomas was one of the ones that sort of drove that movement. I think in terms of the movement, it was an excellent first step towards getting the attention of the breath community in terms of attempting to standardize techniques and sort of just trying to capture that initial variation across techniques when everyone does the same experiment. I think the utility of the results really depends on how you approach the experiment and what you're expecting from it. So like I say, I think it's a good step in the right direction for standardization. It's not going to solve all of the standardization issues, but it makes sense when you're lacking sort of an internal reference material that everyone can analyze and things like that. And that, I know there has also been developments in that area since that. So yes, it has a use, I think, especially if you're just trying to set up your methods. But I think the important thing to remember there is all the stuff that came before the peppermint protocol that people like Paul sort of made sure were in place. So developing all your SOPs, making sure you're collecting high fidelity data in the first place. I think all of that comes before. So if you just do the peppermint experiment and expect everything to be standardized, it's never going to happen. You have to have all your methods in place first, and then you can use something like that to sort of get an idea of where you stand in terms of what other people are doing.
Yeah, I don't know if anyone else is familiar with it. I think one of the questions I had, Mike, was there's because the substrates themselves are being metabolized in the liver, there's inherently going to be a lot of biological variation there. So you can't isolate that very easily. So even if you use the same substrates, we know from administering fixed dose of pure limiting, for instance, you get just huge amounts of biological variation. Anyway, and that's what allows you to separate out disease versus healthy controls.
No, I think it'd be much. So I think then a step on from that then would be people using the same, where possible, the same samples from the same subjects, but then obviously you're limited based on, depending what sample you're using, all sorts of different things. Yeah, that inherent biological variation turned out to be absolutely massive for that particular substrate. Whether I don't think that was as obvious as it was until people started doing the experiment. And so, okay, what can be changed from that? But I think it sort of called to arms everyone's approach to sort of doing breath analysis.
And as I say, it's definitely, if you're looking to have something that you know is definitely going to be there, so it's a way to test your setup, it's something that's great to check. We'll move on and talk a little bit about the projects looking at healthy human VOCs. And again, I think what's quite exciting with Brooke, Ethan, and Mangi, these are all involved in the project to try and build this type of data set and this type of knowledge. And the question that we posed to the attendees was, let me just find it quickly. How important do you think it is to do this work, so to identify and quantify VOCs in nominally healthy subjects and to make it available to the research community? So yesterday, so it's a score of 1 to 5, where 5 is critically important for the field. Yesterday it scored 4.42 for the attendees yesterday, and today it scored 4.5. And I think some of the comments were fascinating. So without a healthy control comparator set, how can you evaluate disease states? Building databases is essential. To understand deviation from healthy, we need to understand the healthy range. It's important to establish normal VOC breath profile for future comparisons with abnormal. It's imperative to establish the range limits of variability within the normal VOC breath profile. And a lot of the comments basically go on a lot like that. So I think what's great is that it seems that the work that you all are doing is eagerly needed by all of us working within breath research. I was wondering if we could start with you just giving a bit of a summary of where you are so far with the various research strands. Maybe, Brooke, we could start with you.
Sure. So we, at the stage in our project right now, we have collected all of our samples from our cohorts, and we're in the process of analyzing them. So with 1,700 samples, it takes a while. So we're working through those. And of course, instruments decide that they don't want to work for a little while, and then they come back and they have their own personalities. But yeah, so we're doing data acquisition, and then from the data that we've already gathered, we're starting to analyze that. We don't want to wait until the end and then figure out that we should have done something different, right? So we're doing kind of stepwise approach as far as the data and statistical analysis. So aligning data sets, looking at trends, looking at things that appear to be enriched in breath as compared to background, as I described before. And then we're also working on a kind of collaborative effort between the three of us, taking in data from all three of our groups and trying to understand what is in common between all three of us. So, at the end of our projects, we'll each have our individual kind of lists of things, or, our understanding of normal breath. And then we'll also have a combined, kind of think of it as a Venn diagram, I guess, where we'll have things that we're all finding in our different approaches and different cohorts and all those different variables.
I think that loops back to the point you made earlier. If you triangulate from different directions, there'll be some commonality of things where the different platforms can see those compounds, and then new things that will be seen only by platforms of different capabilities. Mangi, is there anything you want to kind of add there in terms of what you're seeing so far?
No, Brooke covered our combined effort. We have collected the samples, ran the samples through our GCMS and nanosensor array. We have the data. Now we are trying to ID the VOCs and trying to also quantify the range of VOCs that are found in intra-subject, also inter-subject, so we have longitudinal data from single individual subjects, and then we have close to 150 subjects that we have collected breath from. So both inter and intra-subject analysis is being done, and then working with Brooke and Ethan to see how our data can be, comprehensive with respect to the overall goal of DTRA in terms of getting a healthy baseline database and prototype.
And Ethan, just kind of following up with you on that, I think one of the lines I read most frequently in breath research papers is there's over 1,000 VOCs on breath. And I'm like, I would love to know where that originated and what those 1,000 VOCs are that are actually on breath. So building on the original question, do you have a sense of, how, given the challenges dealing with background, etc., do you have a sense of how many VOCs you think are on breath using, your approach of Cesium Orbitrap and then threading and some of the other approaches as well?
Yeah, absolutely. I always reference this review paper. We went back and to have some sort of a benchmark, pulled out every single VOC that was listed in this review and how many were found in breath. And about 75% of those had one reference, and I think about 50% of those were actually from a personal communication. So it kind of starts to throw a wrench in the system a little bit. I think it's pretty reasonable, at least from our side. We're always going to be looking for what percentage of our samples was this VOC seen in, and I know that you're obviously very familiar with that. I think we go all the way down even to like a couple of percent. Sort of going back to what Michael said, there's so many times that there's certain things that you expect to see because maybe there's a biochemical pathway you're familiar with, but it's so often that there could be compounds or features that maybe you weren't expecting that might make sense in that data set. At least for us, I can say butylated hydroxytoluene was one that we weren't expecting, but sort of makes sense from sort of that food perspective. So at least from us, our first cohort is going to be published in JBR pretty soon, hopefully. And from that, we had about 50 VOCs that we looked at. And that was just based off of the proportion of samples that they were seen in. So I think that kind of goes back to what we were talking about with the, how do you report something that's been identified? I think whenever you have those really stringent requirements for what identification means, that's really going to tell you how many compounds you're seeing in a given cohort. So yeah, for sure.
I think that's interesting. Seeing when you think about nominally healthy subjects and getting reference ranges, one of the potential objections can be, well, what if something is very infrequent? So, may only show up in a disease cohort, for instance. So it sounds like what you're saying there, Ethan, is that, even if something is present in a relatively small subset, that is still potentially quite important. I think when we had a discussion ahead of this call, the example of methane came up actually. So, methane is on breath at quite high concentration, single digit ppm, tens of ppm, but only in a subset of subjects. So I'm wondering if we could explore that a little bit more, for these VOCs in healthy subjects. What do we think about the fraction of the population that they need to show up in? Maybe we could start with you, Mike, on that one.
Yes, so that question, just to comment on that question of 1,000 VOCs on breath, I too have searched for this because as someone that uses GCGC, and so first when setting up a breath method of GCGC, we should be able to see pretty much everything that's on there if we're going to see it. And I was not seeing that per patient. And I think I can track it back to there's two papers I have in mind that were sort of original people using similar techniques. I think in Linus Pauling's original paper, he reported 250. that's probably much more in line of what people would see. You do see up to sort of 500, 700 sometimes, depending on the person. But I think that depends, one, on what you call a VOC or a chromatographic feature or a mass spectral feature, and two, I think that's what you're expecting on a single breath or also over an entire population. So there could be 1,000 different across a larger sample set that has that sort of natural variation, but within a single breath, I think I'm yet to see that level of complexity. I've even seen sort of 2,000, 3,000 quoted. So yeah, just in terms of what the prevalence of the VOCs, I think that's a tricky one, because that comes back to the mechanism, as you say. They will have a natural prevalence within the population anyway, which I guess is why you're doing the VOC atlas, is to get a better understanding of that. So yeah, I don't know. It's hard to. I don't know if the question was what sort of prevalence you would expect, but I think that's very difficult to answer without sort of looking specifically at the VOC. You give a very specific example of methane. Again, I think it depends if you're looking at one or two compounds of interest, or if you're looking at a broader range as well.
Yeah, no, because I think it's yeah. So the notion of whether or not you have something that is truly unique, or if you look in standard blood-based metabolomics, more often not, you have a level present of the metabolites, and you have the normal healthy range, and look for deviations above that range. So it feels like breath should probably, it's volatile metabolomics, so you should probably be a bit like that. So actually the argument that volatiles that are present in the breath of most subjects are probably the most valuable things, because you can look for deviation beyond that. And that's why when we think about the criteria for whether or not someone is on breath, we say it must be above the background, but also must be present in 50% of the population. But that's a bit of an arbitrary number. Why not 10? Why not 70? So it's a bit hard, and it goes back to the standardization side of things a little bit.
So we set actually a higher frequency of observation. So we set about an 80% threshold per group. So not across all of them, but per the group of interest. So if there is something that's not unique, but more indicative of one, that would then maybe be absent from another, or severely reduced, then so yeah, we use a threshold of about 80%.
Interesting. And you got time probably just for one more question. It kind of relates back to the VOC atlas and VOCs in healthy human subjects. Do we have enough kind of information now to get a bit of a sense of where the majority of VOCs are originating from? So is it the airways, GI, liver? So do we have enough information now to make a bit of a prediction about what application areas may best fit the use of breath? And infectious disease obviously is another kind of area where I think you can make a very strong biological argument. So maybe I'll start with Mangi on that one. From what we know on healthy subjects, what's your kind of predictions for where breath tests will be deployed?
Yeah, so basically the VOCs that we saw mostly consist of terpenes, terpenoids, carbonyls, aromatics, and other hydrocarbons that were present, like how Mike said, above 50% of the subjects that we analyzed from. And then we also have, as I said, some disease data from COVID, that is viral infection, and from, there is a lot of database out there for lung cancer, and some of these VOCs correlates to those conditions. So it's very hard to say where we have not done the metabolic studies yet. We are in that process to first, if we can properly ID the VOCs, and then go back and work with our biochemistry team to really find the pathways. But that's an important question to understand where they are generating, where these VOCs are generating from.
Brooke, any predictions you want to make?
Sure. I'm a little biased because I'm a microbiologist, so I just kind of focus in on, infectious disease to me seems like a place, and specifically respiratory infection, because you not only have, the immune response that's going to change things, but also potentially even volatiles that are released from, bacterial infection, for example. And so I think that there's a lot of potential there to kind of see how those different factors come together could be really powerful for diagnostic for infectious disease.
Yeah, I'm with you on infection. Like looking at even fungal infections, some of the potential for the unique volatiles there, I think, is really quite exciting. Ethan, any kind of predictions from you?
Yeah, I really feel like the field is sort of standing on the precipice of this, like understanding the bacterial contribution to that VOC profile and knowing how different the bacteria are from your mouth to your airways to your GI tract. There's just so much potential there, especially for, being able to input certain compounds that might not be VOCs, and having a VOC on the back end is unbelievably powerful.
Yeah.
So that to me is really, really exciting, and starting to sort of probe, like Brooke was talking about, how those bacteria are responding and how we're able to look for VOCs and semi-VOCs and things like that from bacteria is absolutely fascinating.
Yeah, and I agree. Putting substrates in to target them and release unique volatiles gets around a lot of the challenges of background and the like. Mike, obviously we heard about some of your fantastic work yesterday. Do you want to have the last word on this?
I agree. I also have an interest in infection that I'm starting off doing some more in vitro stuff, because we sort of went straight into the human stuff, and bacteria are much more easier to replicate and sort of do the validation on, and think not easier, but a few less ethical constraints and things like that. So, yeah, no, I'm totally on board. I'm on the infection train. Yeah.
Excellent. Well, I want to say a massive thank you to everyone for your contributions today. And again, I think I'll share the poll results with you all and with everyone that's attending the conference. I think the message that's coming through loud and clear is this work is important. It needs to be done. We need to characterize what's on breath in healthy subjects to define those normal ranges, and then that can be applied when you look in different disease populations and making that data available as well. So I know everyone on the call's working on that. That's certainly something that we're doing at Alston as well. Keen to share our results, and we've got poster presentations on our Atlas work as well. So I think hopefully as a field we'll be able to crack this problem together and get that data out there for everyone that comes after us to do research. So thank you again to the panel for your wonderful contribution today.